G.E.N.I.E, and I am here to help you know secrets of nucleotide sequences.
And we are
the Anaconda.team.
And we are here
to share the journey of how we made
genetic code,
electronic decoder,
nucleotide,
information extractor,
G.E.N.I.E.
Okay, so the user can input a data either in the form of a txt file, or he or she can directly copy, paste, or type the text in the box. So the data can contain ATGC sequence, any other information, for example, white spaces or small ATGC, they are going to be eliminated. Once the sequence is entered, the user needs to checkbox the options or select the options from the checkbox. So the options we provide are like the nucleotide count, the molecular weight, the restriction enzyme sites. The user can also search for specific gene sequence and also the complementary strands.
So for the protein translation portion, when the user inputs the sequence is divided into groups of 3, because a code of 3 will code for one amino acid. Similarly, then each group of 3 is checked at the back end for whatever amino acid it matches for, and then an amino acid chain is produced, and that gives you the protein translation portion. In addition to that, we also tell where can this protein be found in a cell. For example, if the resultant protein is KDEL, that means it's present in the endoplasmic reticulum. Similarly, it tells us for quite a few other organelles, and we'll be getting more in the future as well. And it also tells which protein it is for some of the more commonly used proteins, such as hemoglobin, etc.
The sequence length gives the length of the sequence by counting the number of nitrogen bases present in the given input sequence. And in case of nucleotide count, it counts each nitrogen base present in it and gives its output. In case of molecular weight, it finds the molecular weight of whole sequence with the input sequence, its complementary, and as well as the complementary and input sequence double-stranded DNA. Also it also gives output for complementary strand, which is like the A is complementary to T and G is complementary to C. In case of restriction enzymes, it searches for restriction enzyme sites on the given input, and one by one taking one nitrogen base into consideration, and after finding its position, it gives the restriction enzyme. And in the tables option, we are giving what all restriction enzymes are available. So on its basis, if we select one restriction enzyme, it will give its position.
So when the user selects for a specific sequence search, we ask the user for the sequence, and then we traverse through the whole original sequence and find the particular sequence in the forward direction as well as in the reverse direction. So, and if the particular sequence is possible at multiple sites in either of the direction, we provide a list of positions where that specific sequence is located.
Apart from all the functions you've just described that, after the user inputs and what all our main program does, we have also offered a lot of information about biotech and genetics. Our main goal with this was to, educate the beginners, want to find out more about the field, so they can also get theoretical knowledge as well as, apply that knowledge with the functions that we have just described previously.
There are some fundamental concepts in the Biotechpedia that you all know, like what are active sites, what are the amino acid, what is a chromatography, electrophoresis, and all. And there are some common techniques in the Biotechpedia also. Also, some scientific breakthroughs are given, like the Human Genome Project, the CRISPR-based technology, and how the plasmids and restriction enzymes were discovered. And we will also add some more videos in the future too.
So what are you waiting for? Visit our Android app on Play Store today. Link available in the description. Thanks for watching. Have a knowledgeable day.